our first speaker today is Dr. Anand Gururajan. He's a research fellow in the School of Psychology and also at the Brain and Mind Centre, Sydney University. He was awarded the University of Sydney Research Fellowship to investigate the role of RNA and RNA dysregulation in the neurobiology of post-traumatic stress disorder. And Anand is going to speak to us today about epitranscriptomics. He'll have to give me some lessons on how to say that word. A new frontier in molecular psychiatry. So I'd like to hand over to Anand.
Okay, great. I'm assuming everyone can hear me. I'm not sure if they can see me. Great, awesome. Okay, so thanks, Leanne. So as introduced, my name is Anand, and I joined the BMC in 2018 on a fellowship from the DVCR to develop a new research team focused on the epitranscriptome. So for this talk, I'll give you a quick overview of what the epitranscriptome is, why it might be important, the experiments that I did here in the BMC in collaboration with the Garvan, and some of the data that we're planning to submit for a manuscript. Okay, so the epitranscriptome is the collective term used to describe over 100 different types of chemical modifications to RNA molecules. And these are added by RNA binding proteins called writers. They're removed by erasers, and they're detected by readers. And so together, these enzymes and the modifications themselves influence RNA structure and metabolism within the cell. So over the last few years, the functional roles of specific modifications has become clear in the context of various disorders such as cancer, but more recently, they've also been implicated in a number of different neuropsychiatric disorders as well. And one of the hurdles the field has faced is the limited accuracy of the techniques to detect these modifications, but this is about to change, as I'll show you shortly. On the right-hand side, you can see a schematic highlighting the functional role of RNA modifications to newly transcribed messenger RNA. So as previously mentioned, the modifications can influence whether an RNA is actually transported out of the nucleus, whether it's translated into functional protein, how efficient the translation is, how stable it is, and also its structure. So collectively, all of these mechanisms can influence RNA decay, distribution, and function in a manner which contributes to adaptive, experience-dependent gene expression profiles. And we and others have hypothesized that a dysregulation in these mechanisms might contribute to the pathology of psychiatric disorders. So for a long time, the techniques to profile the epitranscriptome have been limited in their specificity and also the resolution. So the game-changing technology arrived more than a decade ago, and it's called Oxford Nanopore. But its application and popularity has only recently took off. Indeed, there has been much done using this technology in the context of COVID-19 research. So the fundamental features of this platform is that RNA or DNA can be directly sequenced by passing the strands through a synthetic pore embedded within an electrified membrane. And as it passes through, each base has its own unique electrical signature, which can then be used to identify the base itself plus any other RNA modifications to that base. So what makes this direct sequencing platform a step above the conventional approaches is that there's a limited number of pre-processing sample steps. So what you see in the data is pretty much likely to be a true reflection of RNA sequence and also the state. So this is cutting edge technology, still quite expensive to use, and there aren't too many places in Australia that use it. But thanks to Twitter and some extensive networking on my part, I was fortunate enough to be introduced to Dr. Aru Davison in 2019 at the Garvan. And he had just set up a nanopore lab at the time and was looking for people to collaborate with to explore the capabilities of this technology. So that's what we did. So in our experiment, we sought to profile the epitranscriptome in a mouse model for aspects of PTSD using a Pavlovian cued fear conditioning paradigm that you can see here. In particular, we focused on the changes to the epitranscriptome that underpin the acquisition of the PTSD-like fear memory in the amygdala, which is a critical structure in this context. So there's a lot of data in this slide here, but I'm just going to walk you through some of the main highlights that I'm going to box in red. So firstly, the acquisition of PTSD-like fear is associated with an increase in the number of RNA modifications in both the male and female amygdala. Secondly, each RNA strand is subject to more than one type of chemical modification, and the numerous modifications are scattered throughout the length of the strand. And one of the interesting hypotheses in the field is that some of these modifications might have a functional impact on RNA on their own, but they might also interact with each other to influence RNA structure and metabolism. So thirdly, the majority of modifications are found at the intersection between the coding sequence and the 3' untranslated region of an RNA strand. So this is a highly active binding site for other post-transcriptional regulators of gene expression, such as non-coding RNAs. So fourthly, very few differentially modified RNAs are also differentially expressed, suggesting an influential expression independent role for epitranscriptional regulation in PTSD, like PTSD acquisition of fear memory. And finally, modified RNAs were enriched for several processes, including mitochondrial metabolism, synaptic activity, and neurotransmission. And all of these processes are known to be involved in the acquisition of fear memory, learning, and also other memory-dependent processes. So to finish off, the data which I presented to you is probably the first chapter in a very long and complex story that's yet to be fully told. Some of the future work that I'd like to do include what is the functional role of specific RNA modifications, and I'm looking to collaborate with researchers across Australia for this. What is the epitranscriptome profile in clinical PTSD? Can peripheral modified RNA serve as diagnostic biomarkers for PTSD? And lastly, are the RNA binding proteins and the enzymes which erase, write, or read these modifications druggable targets? So it goes without saying that RNA research is highly topical right now, and there are a number of companies that are in the races to understand its influence in health and disease, and it's a truly exciting space to be working in. So I'm delighted that the preprint of this work is about to go live, and we plan on submitting it at the same time to a relevant journal, and we hope for a good outcome. I just want to quickly thank all the people who were involved and gave advice on this project. And I also want to quickly wish everyone that's zooming in a happy Diwali. Thanks, Leanne.
Thank you, Anand, for that whirlwind journey through an awful lot of amazing research. I've got one question for you. Is there any evidence to suggest that modified RNAs could be biomarkers for any psychiatric disorders?
Yeah, so there's been one study that was done, that was published, I think 2 years ago now, and they looked at peripheral modified RNAs as being biomarkers for major depression. And they did see, a condition-specific phenotype, but that was a pretty small study, and we would definitely need to kind of increase the numbers and also look at treatment response, whether these modified RNAs are modified by different types of treatments. So there's a lot more work to be done, and that's one of the projects that I'm trying to get funding for. So we'll see how we go next year.
Well, good luck with that. It sounds like extremely worthwhile work, and it's been great to hear from you today.